All right, welcome. Today, we're diving deep into one of the most incredible conflicts in all of biology, this molecular arms race between a virus and its host. And we're going to frame this as a high-stakes heist, where the ultimate prize is the cell's protein-making factory. It's an amazing story of sabotage, subterfuge, and just some brilliant evolutionary engineering. So let's map out our plan. Here's how we're going to break it down. First, we'll start by casing the joint. Getting to know the cellular protein factory itself. Then we'll learn the standard blueprint for how it's supposed to operate. Once we've got that down, we'll dig into how viruses actually pull off this heist, from direct, all-out sabotage to really clever inside jobs and some other advanced hacks. And finally, we'll zoom out and look at the bigger picture, this perpetual evolutionary arms race. Okay, so every great heist starts with a target, right? For a virus, the single most valuable asset inside a host cell is its translation machinery. We're talking about the ribosome. See, a virus is basically just a set of blueprints with no tools. It has to make copies of itself, it needs proteins, it needs enzymes, and to do any of that, it absolutely has to hijack the cell's factory. Now this right here, this gets to the absolute core of the issue. Viruses are what we call obligate intracellular parasites. That's a fancy way of saying they are completely 100% dependent on the inside of a host cell to replicate. They have no machinery of their own to build proteins. None. So they must hijack the host's ribosome. Now think about what that means. Every viral mRNA is in direct competition with thousands of cellular mRNAs, all fighting for access to these factories. To win, viruses can't just compete, they have to cheat. So before a virus can even think about bypassing the system, it's got to understand how it works. So let's take a minute and break down the highly secure standard operating procedure for making a protein inside a eukaryotic cell. So every official blueprint, what we call messenger RNA, in a eukaryotic cell has these really specific security features. At the front, the 5' end, there's a special modified nucleotide called a cap. Think of it like an official seal on a letter. Then at the back, the 3' end, there's a long string of adenine bases. That's the poly A tail. Now, these aren't just for decoration. They're absolutely critical for protecting the message from getting destroyed and, most importantly, for getting it translated. They are the molecular security badges that grant access to the ribosome. The whole process of reading that blueprint happens in 3 main stages. First up is initiation. That's where all the ribosome machinery assembles on the mRNA. Then you've got elongation, the actual building of the protein chain. And finally, termination, where the completed protein is released. And as we're about to see, viruses have evolved some truly sneaky ways to interfere with every single one of these stages. This diagram here gives us a fantastic detailed overview of that entire process. You can see initiation right at the top, all the factors coming together. Elongation is that bit in the middle showing the ribosome just chugging along the mRNA, and termination is there at the bottom. But while viruses can target any of these steps, the main battleground, the primary point of control, is almost always that very first step, initiation. Let's look at why. So why initiation? Well, because it's the most regulated, it's the rate-limiting step, it is the main checkpoint. If a virus can control initiation, it controls the entire factory. I mean, it's no use having blueprints if you can't even get them through the front door, right? So the key takeaway here is that viruses have become absolute master locksmiths of this molecular front door. So who's the gatekeeper at this front door? It's a protein complex called EIF4F, and you can just think of it as the keymaster. It's made of 3 essential parts. You've got EIF4E, which binds directly to that 5' cap. It's the part that physically checks the security badge. Then there's EIF4G, a big scaffolding protein that basically holds everything together. And finally, EIF4A, which is a helicase that unwinds any knots or tangles in the mRNA to clear a path. Together, this complex is essential for standard cap-dependent translation. And this just illustrates how unbelievably efficient the cell makes this whole process. See that poly A binding protein, or PAB? It grabs onto the poly A tail at the back end, but it also interacts with the EIF4F complex at the front end. This physically brings the 2 ends of the mRNA together, forming a closed loop. So why does this matter? It means that as soon as a ribosome finishes translating and pops off the end, it's immediately positioned right next to the start, ready to jump back on and go again. It's a masterpiece of molecular engineering for super fast, continuous protein production. All right, now that we know the factory layout, let's look at the first viral strategy, a direct frontal assault. This isn't subtle. This is all about shutting down the competition by sheer brute force.
Some viruses just go for a complete hostile takeover. Herpes simplex virus, for example, produces an enzyme that's basically a shredder. It just chews up host mRNAs to get them out of the way. But influenza virus, its strategy is even more audacious. It's called cap snatching. It literally steals the 5' caps from host mRNAs and sticks them onto its own viral RNAs. This does two incredible things at once. It destroys the host's blueprints while simultaneously camouflaging its own as legitimate. It's an unbelievably aggressive and effective way to make sure only viral proteins get made.
But, a frontal assault isn't always the best move. Some viruses prefer a much more subtle approach, a classic inside job. Instead of blowing up the front door, they use a secret entrance that nobody else even knows is there. And this slide shows the two mechanisms side by side perfectly. On the left, you've got the normal process we just talked about. The ribosome machinery recognizes that 5' cap. But look at the right. That's the viral trick. The viral RNA has a special, highly structured region called an internal ribosome entry site, or an IRES. This IRES acts like a magnetic landing pad, letting the ribosome assemble directly in the middle of the RNA molecule. It completely bypasses the need for a 5' cap. It is a built-in backdoor. Poliovirus is the absolute master of this strategy, and its plan is just diabolically elegant. First, it makes a viral protease, an enzyme called 2A. Now think of this protease as a tiny pair of molecular scissors. Its job is to find the host's EIF4G scaffolding protein and just snip it. That one little cut completely disables the cell's ability to do normal, cap-dependent translation. The front door is now locked for all host mRNAs. But, and here's the genius part, the poliovirus IRES doesn't need the whole intact EIF4G. It can directly recruit the part that's been cut off along with the ribosome and start its own translation. It sabotages the entire host system and then hijacks the broken pieces for itself. It's brilliant. Okay, so beyond just getting access to the ribosomes, viruses face another huge challenge, their genome size. They have to pack an incredible amount of information into a tiny genetic space. To do this, they've evolved some amazingly clever hacks to make a single piece of RNA do the work of 2 or even 3. Let's look at a few of these. First up, we have something called leaky scanning. So in our cells, the ribosome doesn't just look for any AUG start codon. It's looking for one in an optimal sequence context. Viruses totally exploit this. They can put their first start codon in a really weak context, making it less attractive. This means some ribosomes will start there, sure, but a lot of them will just skip right over it. They leak past it and start translation at a second, stronger start codon further down. And the result? One single mRNA can produce 2 or more totally different proteins. It's a masterclass in genetic economy. A similar trick is reinitiation. In this case, a virus will have a very short little open reading frame, an ORF, at the beginning of its message. After translating this tiny peptide, the ribosome stops, but instead of falling off completely, it's held in place and told to start scanning again. It can then reinitiate translation at a real start codon further downstream. This effectively lets one mRNA be polycistronic, making multiple distinct proteins from a single blueprint. This is normal in bacteria, but it's really unusual in eukaryotes, making it another system that viruses have learned to exploit brilliantly. But maybe the most mind-bending trick of all is ribosomal frame shifting. This is used by retroviruses like HIV and even coronaviruses. The viral RNA has a specific sequence and a kind of structural knot that forces the ribosome to pause and actually slip backwards by one nucleotide. This shifts the entire reading frame. The ribosome keeps on translating, but now it's reading a completely different set of codons, making a large fusion protein that's totally different at its end. It is a stunning way to compress 2 distinct protein coding sequences into the space of one. So, what's the big picture consequence of all these molecular heists and hacks? Well, it's a relentless evolutionary battle. It's been raging for millions and millions of years, and it's shaped both viral and host biology in profound ways. So let's just quickly recap the viral playbook. Viruses win by rewriting the rules. They either demolish the competition through direct sabotage, or they use stealth, bypassing the cell's normal security with their own backdoors like the IRS. And once they're inside, they use these incredibly sophisticated coding tricks like leaky scanning and frame shifting to maximize their output. This constant pressure to innovate is a powerful engine of evolution for both sides of the conflict. And that brings us to our final thought. We've spent this entire time focused on the virus's side of the heist, all its ingenious tools and strategies. But security systems always evolve in response to break-ins. So the question I want to leave you with is this. What countermeasures has the host cell developed to detect and stop these molecular pirates? What does the cell's antiviral security system actually look like? Well, that's a story for another time. Thanks for joining me.